So, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Sharafuddin bin Azmi. I'm from class DHP9 and today I will show you how to we get the recombinant DNA. So, firstly, the first step for recombinant DNA is isolation. So, isolation is where we take the plasmid from the bacteria cell, here the plasmid, and the target DNA from human cell where we isolate it by vitro test. So firstly, we identify the restriction site for at the late Z for restriction enzyme. So for plasmid A, as we can see, we have found the restriction site for enzyme EcoR1, which is GAA TTC CTTAAG. So when we cut the plasmid, it will produce sticky end. So for the second plasmid, we can see we have found the restriction site at the late Z for BAMH1, which is GGA TCC, and by the palindromic sequence we got CCTA-GG. So the BAMH1 also, when we cut the end at the plasmid, we will get the sticky end too. So for the last plasmid, which is plasmid C, we have found the small one, which is when we cut the plasmid, so we will get the blonde end at the late Z. So the next one, we should identify the restriction site for late Z at the DNA. The example is human DNA. So first, for DNA 1, we have identified the restriction site for small one, which is GGGCCC. And for DNA 2, the restriction enzyme we found at the late Z is GAA-TTC, which is the EcoR1. The EcoR1. So the last DNA, which is DNA 3, the restriction enzyme that we found is GGA-TCC, which is for BAMH1. So as you can see, I have marked the restriction site for the EcoR1 restriction enzyme here at the late Z for plasmid A. So after we cut here, it will open and divide into two. So at the open place, we will cut the EcoR1 at the human DNA and we combine it by glue it together, so we can have the recombinant DNA. So I have first obtained my first recombinant DNA, which is from the EcoR1 restriction enzyme. So as you can see, this is a plasmid and this is from human foreign DNA, as you can see. So I obtained my recombinant DNA by the second step, which is the cleave and cut the DNA from human fragment and from the plasmid with restriction enzyme with the same size, because we want to obtain the compatible recombinant DNA. So this is my plasmid B. I will cut here. If you can see, I have marked here with my green pen for the BAMH1 restriction enzyme. So I will cut it and do the process insertion, which is I will insert the BAMH1 restriction enzyme at the human DNA to the plasmid, so we can obtain the recombinant DNA. So as you can see here, I finally obtained the second recombinant DNA, which is here, which is I cut using the BAMH1. So the final step is insertion, which insert the here green color here foreign DNA from the human fragment into the plasmid to form the recombinant DNA. So this is my third plasmid. So as you can see, I have marked here with my green pen. So I will cleave and cut it with using small one. So I will to obtain the blunt end. So it will cut here. So this restriction enzyme, the restriction site for the SMA1 restriction enzyme is CCC-GGG, and by palindromic sequence GGG-CCC. So this is my last recombinant DNA that I have obtained, which is here. You can see this is the restriction site from the human DNA, and the white color is from the plasmid from the bacteria cell. So this is the insertion process, which is I insert the human DNA into the bacteria cell, that is plasmid. So as you can see, I cut this using the same restriction enzyme, which is SMA1. Look, CCC-GGG, and the palindromic sequence GGG-CCC. So that's the end of this activity on how to obtain the recombinant DNA. So that's all for it, and thank you for watching this video, and wish me good luck. Thank you.
House music.